Bisulfite sequencing is the use of bisulfite treatment of DNA to determine its pattern of methylation. DNA methylation was the first discovered epigenetic mark, and remains the most studied. In animals it predominantly involves the addition of a methyl group to the carbon 5 position of cytosine residues of the dinucleotide CpG, and is implicated in repression of transcriptional activity. Treatment of DNA with bisulfite converts cytosine residues to uracil, but leaves 5-methylcytosine residues unaffected. Thus, bisulfite treatment introduces specific changes in the DNA sequence that depend on the methylation status of individual cytosine residues, yielding single-nucleotide resolution information about the methylation status of a segment of DNA. Various analyses can be performed on the altered sequence to retrieve this information. The objective of this analysis is therefore reduced to differentiating between single-nucleotide polymorphisms resulting from bisulfite conversion. Methods: Bisulfite sequencing applies routine sequencing methods on bisulfite-treated genomic DNA to determine methylation status at CpG dinucleotides. Other non-sequencing strategies are also employed to interrogate the methylation at specific loci or at a genome-wide level. All strategies assume that bisulfite-induced conversion of unmethylated cytosines to uracil is complete, and this serves as the basis of all subsequent techniques. Ideally, the method used would determine the methylation status separately for each allele. Alternative methods to bisulfite sequencing include combined bisulfite restriction analysis and methylated DNA immunoprecipitation. Methodologies to analyze bisulfite-treated DNA are continuously being developed. To summarize these rapidly evolving methodologies, numerous review articles have been written. The methodologies can be generally divided into strategies based on methylation specific PCR, and strategies employing polymerase chain reaction performed under non-methylation specific conditions. Microarray-based methods use PCR based on non-methylation specific conditions also. Non-methylation specific PCR-based methods equals direct sequencing. The first reported method of methylation analysis using bisulfite-treated DNA utilized PCR and standard deoxynucleotide DNA sequencing to directly determine the nucleotides resistant to bisulfite conversion. Primers are designed to be strand-specific as well as bisulfite-specific, flanking the methylation site of interest. Therefore, it will amplify both methylated and unmethylated sequences, in contrast to methylation-specific PCR. All sites of unmethylated cytosines are displayed as thymines in the resulting amplified sequence of the sense strand, and as adenines in the amplified antisense strand. This technique required cloning of the PCR product prior to sequencing for adequate sensitivity, and therefore was a very labor-intensive method unsuitable for higher throughput. Alternatively, nested PCR methods can be used to enhance the product for sequencing. All subsequent DNA methylation analysis techniques using bisulfite-treated DNA is based on this report by Fromo et al. Although most other modalities are not true sequencing-based techniques, the term bisulfite sequencing is often used to describe bisulfite conversion DNA methylation analysis techniques in general. Pyro sequencing. Pyro sequencing has also been used to analyze bisulfite-treated DNA without using methylation-specific PCR. Following PCR amplification of the region of interest, pyro sequencing is used to determine the bisulfite-converted sequence of specific CpG sites in the region. The ratio of C to T at individual sites can be determined quantitatively based on the amount of C and T incorporation during the sequence extension. The main limitation of this method is the cost of the technology. However, pyro sequencing does well allow for extension to high-throughput screening methods. A further improvement to this technique was recently described by Wang et al., which uses allele-specific primers that incorporate single-nucleotide polymorphisms into the sequence of the sequencing primer, thus allowing for separate analysis of maternal and paternal alleles. This technique is of particular usefulness for genomic imprinting analysis. Methylation-sensitive single-strand conformation analysis. This method is based on the single-strand conformation polymorphism analysis method developed for single-nucleotide polymorphism analysis. SSCA differentiates between single-stranded DNA fragments of identical size but distinct sequence based on differential migration and non-denaturating electrophoresis. In MIS-SSCA, this is used to distinguish between bisulfate-treated, PCR-amplified regions containing the CpG sites of interest. Although SSCA lacks sensitivity when only a single nucleotide difference is present, bisulfate treatment frequently makes a number of C to T conversions in most regions of interest, and the resulting sensitivity approaches 100%. MIS-SSCA also provides semi-quantitative analysis of the degree of DNA methylation based on the ratio of band intensities. However, this method is designed to assess all CpG sites as a whole in the region of interest rather than individual methylation sites. High-resolution melting analysis, a further method to differentiate converted from unconverted bisulfate-treated DNA, is using high-resolution melting analysis, a quantitative PCR-based technique initially designed to distinguish SNPs. The PCR amplicons are analyzed directly by temperature ramping and resulting liberation of an intercalating fluorescent dye during melting. The degree of methylation, as represented by the C to T content in the amplicon, determines the rapidity of melting and consequent release of the dye. This method allows direct quantitation in a single tube assay, but assesses methylation in the amplified region as a whole rather than at specific CpG sites. Methylation-sensitive single-nucleotide primer extension, MIS-SNUPE, employs the primer extension method initially designed for analyzing single-nucleotide polymorphisms. DNA is bisulfate converted, and bisulfate-specific primers are annealed to the sequence up to the base pair immediately before the CpG of interest. The primer is allowed to extend one base pair into the C using DNA polymerase terminating dideoxynucleotides, and the ratio of C to T is determined quantitatively. A number of methods can be used to determine the C-T ratio. At the beginning, MIS-SNUPE relied on radioactive ddNTPS as the reporter of the primer extension. Fluorescence-based methods or Pyro Sequencing can also be used. However, matrix-assisted laser desorption ionization time-of-flight mass spectrometry analysis to differentiate between the two polymorphic primer extension products can be used, in essence, based on the good assay designed for SNP genotyping. Ion pair reverse phase high-performance liquid chromatography has also been used to distinguish primer extension products. Base-specific cleavage MALDITOF, a recently described method by Eric et al., further takes advantage of bisulfate conversions by adding a base-specific cleavage step to enhance the information gained from the nucleotide changes. By first using in vitro transcription of the region of interest into RNA, RNA-seq can be used to cleave the RNA transcript at base-specific sites. As RNA-seq cleaves RNA specifically at cytosine and uracil ribonucleotides, base specificity is achieved by adding incorporating cleavage-resistant DTTP when cytosine-specific cleavage is desired, and incorporating DCTP when uracil-specific cleavage is desired. The cleaved fragments can then be analyzed by MALDITOF. Bisulfate treatment results in either introduction removal of cleavage sites by C to U conversions or shift in fragment mass by G to A conversions in the amplified reverse strand. C-specific cleavage will cut specifically at all methylated CpG sites. By analyzing the sizes of the resulting fragments, it is possible to determine the specific pattern of DNA methylation of CpG sites within the region, rather than determining the extent of methylation of the region as a whole. This method demonstrated efficacy for high-throughput screening, allowing for interrogation of numerous CpG sites in multiple tissues in a cost-efficient manner. Methylation Specific Pcr . This alternative method of methylation analysis also uses bisulfate-treated DNA but avoids the need to sequence the area of interest. Instead, primer pairs are designed themselves to be methylated specific by including sequences complementing only unconverted 5-methyl cytosines, or, on the converse, unmethylated specific. complementing thymines converted from unmethylated cytosines. Methylation is determined by the ability of the specific primer to achieve amplification. This method is particularly useful to interrogate CpG islands with possibly high methylation density, as increased numbers of CpG pairs in the primer increase the specificity of the assay. Placing the CpG pair at the free end of the primer also improves the sensitivity. The initial report using MSP described sufficient sensitivity to detect methylation of 0.1% of alleles. In general, MSP and its related protocols are considered to be the most sensitive when interrogating the methylation status at a specific locus. The Methylite method is based on MSP, but provides a quantitative analysis using quantitative PCR. Methylated specific primers are used, and a methylated specific fluorescence reporter probe is also used that anneals to the amplified region. In alternative fashion, the primers or probe can be designed without methylation specificity if discrimination is needed between the CpG pairs within the involved sequences. Quantitation is made in reference to a methylated reference DNA. A modification to this protocol to increase the specificity of the PCR for successfully bisulfite-converted DNA uses an additional probe to bisulfite unconverted DNA to quantify this non-specific amplification. Further methodology using MSP-amplified DNA analyzes the products using melting curve analysis. This method amplifies bisulfite-converted DNA with both methylated specific and unmethylated specific primers, and determines the quantitative ratio of the two products by comparing the differential peaks generated in a melting curve analysis. A high-resolution melting analysis method that uses both quantitative PCR and melting analysis has been introduced, in particular, for sensitive detection of low-level methylation. Microarray-based methods Microarray-based methods are a logical extension of the technologies available to analyze bisulfite-treated DNA to allow for genome-wide analysis of methylation. Oligonucleotide microarrays are designed using pairs of oligonucleotide hybridization probes targeting CpG sites of interest. One is complementary to the unaltered methylated sequence, and the other is complementary to the C to U converted unmethylated sequence. The probes are also bisulfite specific to prevent binding to DNA incompletely converted by bisulfite. The alumina methylation assay is one such assay that applies the bisulfite sequencing technology on a microarray level to generate genome-wide methylation data. Limitations 5-hydroxymethylcytosine bisulfite sequencing is used widely across mammalian genomes, however complications have arisen with the discovery of a new mammalian DNA modification 5-hydroxymethylcytosine. 5-hydroxymethylcytosine converts to cytosine 5-methylsulfonate upon bisulfite treatment, which then reads as a C when sequenced. Therefore, bisulfite sequencing cannot discriminate between 5-methylcytosine and 5-hydroxymethylcytosine. This means that the output from bisulfite sequencing can no longer be defined as solely DNA methylation, as it is the composite of 5-methylcytosine and 5-hydroxymethylcytosine. The development of TET-assisted bisulfite sequencing by Chuani at the University of Chicago is now able to distinguish between the two modifications at single base resolution. Incomplete conversion bisulfite sequencing relies on the conversion of every single unmethylated cytosine residue to uracil. If conversion is incomplete, the subsequent analysis will incorrectly interpret the unconverted unmethylated cytosines as methylated cytosines, resulting in false positive results for methylation. Only cytosines in single-stranded DNA are susceptible to attack by bisulfite, therefore denaturation of the DNA undergoing analysis is critical. It is important to ensure that reaction parameters such as temperature and salt concentration are suitable to maintain the DNA in a single-stranded conformation and allow for complete conversion. Embedding the DNA in agarose gel has been reported to improve the rate of conversion by keeping strands of DNA physically separate. Degradation of DNA during bisulfite treatment A major challenge in bisulfite sequencing is the degradation of DNA that takes place concurrently with the conversion. the conditions necessary for complete conversion, such as long incubation times, elevated temperature, and high bisulfite concentration, can lead to the degradation of about 90% of the incubated DNA. Given that the starting amount of DNA is often limited, such extensive degradation can be problematic. The degradation occurs as depurinations resulting in random strand breaks. Therefore, the longer the desired PCR amplicon, the more limited the number of intact template molecules will likely be. This could lead to the failure of the PCR amplification, or the loss of quantitatively accurate information on methylation levels resulting from the limited sampling of template molecules. Thus, it is important to assess the amount of DNA degradation resulting from the reaction conditions employed, and consider how this will affect the desired amplicon. Techniques can also be used to minimize DNA degradation, such as cycling the incubation temperature. Other concerns. A potentially significant problem following bisulfate treatment is incomplete desulfonation of pyrimidine residues due to inadequate alkylization of the solution. This may inhibit some DNA polymerases, rendering subsequent PCR difficult. However, this situation can be avoided by monitoring the pH of the solution to ensure that desulfonation will be complete. A final concern is that bisulfate treatment greatly reduces the level of complexity in the sample, which can be problematic if multiple PCR reactions are to be performed. Primer design is more difficult, and inappropriate cross-hybridization is more frequent. Applications: genome-wide methylation analysis. The advances in bisulfate sequencing have led to the possibility of applying them at a genome-wide scale, where, previously, global measure of DNA methylation was feasible only using other techniques, such as restriction landmark genomic scanning. The mapping of the human epigenome is seen by many scientists as the logical follow-up to the completion of the Human Genome Project. This epigenomic information will be important in understanding how the function of the genetic sequence is implemented and regulated. Since the epigenome is less stable than the genome, it is thought to be important in gene-environment interactions. Epigenomic mapping is inherently more complex than genome sequencing, however, since the epigenome is much more variable than the genome. Only a unregistered trademark S epigenome varies with age, differs between tissues, is altered by environmental factors, and shows aberrations in diseases. Such rich epigenomic mapping, however, representing different ages, tissue types, and disease states, would yield valuable information on the normal function of epigenetic marks as well as the mechanisms leading to aging and disease. Direct benefits of epigenomic mapping include probable advances in cloning technology. It is believed that failures to produce cloned animals with normal viability and lifespan result from inappropriate patterns of epigenetic marks. Also, aberrant methylation patterns are well characterized in many cancers. Global hypermethylation results in decreased genomic stability, while local hypermethylation of tumor suppressor gene promoters often accounts for their loss of function. Specific patterns of methylation are indicative of specific cancer types, have prognostic value, and can help to guide the best course of treatment. Large-scale epigenome mapping efforts are underway around the world and have been organized under the Human Epigenome Project. This is based on a multi-tiered strategy, whereby bisulfate sequencing is used to obtain high-resolution methylation profiles for a limited number of reference epigenomes, while less thorough analysis is performed on a wider spectrum of samples. This approach is intended to maximize the insight gained from a given amount of resources, as high-resolution genome-wide mapping remains a costly undertaking. Gene set analysis has been shown to be severely biased when applied to high-throughput methylation data. It has been suggested that this can be corrected using sample label permutations or using a statistical model to control for differences in the numbers of CpG probes/CpG sites that target each gene. Oxidative bisulfate sequencing, 5-methylcytosine and 5-hydroxymethylcytosine both read as a C in bisulfate sequencing. Oxidative bisulfate sequencing is a method to discriminate between 5-methylcytosine and 5-hydroxymethylcytosine at single base resolution. The method employs a specific chemical oxidation of 5-hydroxymethylcytosine to 5-formylcytosine, which subsequently converts to uracil during bisulfite treatment. The only base that then reads as a C is 5-uromethylcytosine, giving a map of the true methylation status in the DNA sample. Levels of 5-urohydroxymethylcytosine can also be quantified by measuring the difference between bisulfite and oxidative bisulfite sequencing. See also, reduced representation bisulfite sequencing. References. External links, Bisulfite Conversion Protocol, Human Epigenome Project, data by the Sanger Institute, the Epigenome Network of Excellence.